So as you can see that in the last class we discussed about how we can use or how we can manipulate the amino acid structure and function, or not the function, the amino acid structure, both the physical structure and the chemical structure, for prediction of this particular entire protein structure. Okay, because whatever we do, the servers and the algorithms, they actually use, now there are various methods of protein structure prediction that we're going to see today next. So one was actually the homology modeling, then we have the ab initio method, then we have protein threading and all. So every time that this algorithm and the server that they have, what they do is they actually take a clue from the already solved protein structure. Now those protein structure can be solved by NMR spectroscopy or they can be solved by X-ray crystallography. But they take the clue from there and they use the amino acid zone chemical structure to predict the entire protein structure. Now there are actually two folds of protein structure that we know. One fold is actually the secondary structure and then we have the 3D structure. So secondary structure prediction is very important. Why? Because it gives you the idea about which part is alpha helix, which part is beta sheet, and which part is the random coil structure and all. Okay, so but how do they do that? So that's what we actually discussed the previous day. We discussed that there are few amino acids that have high beta sheet forming propensity, and there are few amino acids that have high alpha helix forming propensity, and both of these amino acid, the structure, the server actually finds out, looks for the amino acid that are present in this particular rich areas, and they assign the particular domain according to that. So today we're going to take a look into one of the server for predicting this particular uh, secondary structure. Okay, so the first, just simply click on the link that I have sent you. So the first, that this is the Chow and Fastman secondary structure prediction server. So what this does is this actually simply predicts the secondary structure of the amino acid sequence. Find it, it is not giving you the entire 3D structure of the protein. It is only showing the secondary structure of the protein. That means it's only going to say which amino acid has been assigned which part of that particular, which secondary structure. Okay, so it is very easy to use. What we have to do is we have to simply upload the or paste the protein sequence in FASTA format. So I have already pasted a sequence. Okay, I have already discussed and found a particular protein sequence for this particular thing, and this is a hexokinase. I know you all, I think you all know what is hexokinase, right? Hexokinase is the protein that is used in the glucose metabolism, right? It is used in the glycolysis pathway.
Glycolysis.
Yes. So I've used this Homo sapiens, simply that particular hexokinase pathway, and simply you have to paste the entire sequence. Now, what is FASTA sequence? The FASTA format starts with the greater than sign, and then you have to write down the name of the protein or whatever, you can write it down. But starting from the next line, you have to give the single letter amino acid code. Single letter amino acid code in capital letter. Okay, so this is what I've done. And after that it is done, we have to simply click in predict, and then it's going to start predicting the secondary structure. So what are the information that we get? So first we have this is the entire just a minute. Okay, so this is the entire target sequence that we have, and these numbers only number the amino acid number. So this is the 10th amino acid, the 20th amino acid, 30th amino acid. It has been written in such a format so that it is easier for one to keep the track of the entire sequence. And this is the entire secondary structure. And here the red actually indicates the helix, the green indicates the beta sheet, and the blue indicates the turns, and the yellow indicates the random coil structure. So if you see the secondary structure prediction, what it does, this is the entire sequence, and first it gives you the helix forming propensity. Now it is obviously trying to predict the helix forming propensity of all these amino acids, and according to the helix forming propensity, it says that this entire stretch has the helix forming propensity, whereas this particular stretch has the helix forming propensity. But not only that, if you cross check with the beta sheet forming propensity, the server also allocates that this particular area starting from here to here, this particular domain also has the propensity to form a beta sheet, which is indicated by this E, letter E. The E denotes the beta sheet, okay, and the H denotes the alpha helix. Now, so both of this region is assigned the alpha helix as well as the beta sheet. But what is going to happen if we mix it up? And considering the entire protein structure, this particular server allocates alpha helix region till this amino acid alpha helix region, and this entire domain LLAY, this entire domain the beta sheet region. Okay, now you might say that why this was actually assigning alpha helix here, it was assigning beta sheet here. So how could we know that this part is beta sheet, this part is alpha helix? What it does is actually, after it predicts the entire sequence, they try to fold the sequence on their own. Okay, and upon folding, sometimes they find that there are certain region where the beta sheet propensity is not there because the protein is folding at that branch. And when the protein is folding at a particular branching point, then it is easier for that to form a alpha helix or a beta sheet. So that's why it gives you the final structure is given by this colored letter. Okay, this colored letter gives you the final structure. So if you simply see it down, you get to see the entire secondary structure of the protein, and you get to have the idea that which part is forming alpha helix, which part is forming beta sheet. And again, if you simply scroll down, the entire structure is given in that way. And if you come down at the end, you are going to find out the total alpha helix forming percentage. Here you could see that there are total 686 residues that are forming alpha helix, 431 residues that are forming beta sheet, and there are 116 residues that are participating in random coils or in the turns. And with respect to percentage, that gives rise to 74.8% of the helix, 47% to the beta sheet, and 12.6% to the turns. Now the question is, you might ask that why do I want to know about the secondary structure and not the 3D structure, right? Now can you tell me that what is the utility of having knowing a secondary structure rather than finding the 3D structure?
We can have an assumption if the protein transmembrane protein or not transmembrane protein, if the protein is soluble or not. Like
very good, absolutely right. Absolutely right. Before forming the 3D structure, we could have an assumption that which part is going to have which structure. Okay, and there is also another important source to this. The important source is, in case of the drug designing, the one who uses the drug designing part, or they use the CAD programming for the computer assisted drug designing, it is very important to know where your molecule might bind to or which area of the protein that you could target. Now you see the one that is forming alpha helix and beta sheet, the region of the protein that has a proper secondary structure, that is highly stable. That region is highly stable. Why? Because they have a proper structure. And alpha helix and beta sheet, they are not prone to be broken down easily because they are stabilized by formation of several hydrogen bonds. So that particular domain is highly structured. Whereas the random coiled part, the part with the random coil or which are the turns, they don't have any proper structure. Why? They are simply moving around. They are simply like that particular thread. If you put a thread in the water and if you simply move the water, the thread is going to take different structure. The same thing happens with the random coil nature of the coils of the protein also. According to the our blood is actually a colloidal solution. So obviously there are random molecules that are coming and binding with protein, that are pushing the protein, and with each random collision, the coiled structure or that particular domain of protein changes. So if I want to target a drug to a protein, I should obviously choose the structured area, because I know that particular domain is not going to change the structure. And for proper binding, we need a proper structure of protein, because obviously when we want to develop a ligand or a molecule or a drug, we want that molecule to be highly specific to that particular protein. And that is why we use SBDD, that is structure based drug design. We use the structure of the protein so as to so that we can develop a complementary structured ligand. So while we target that particular ligand to this particular alpha helical region or beta sheet region, we know that that particular domain has a proper structure and can bind to that. But if somehow, suppose I want to target the protein to this region, I want to target the protein to this region, I find that this particular domain is very good, I target it. But at the end, I could see that this particular domain forms the random coil structure and turns. So I know that they don't have a proper structure. The ligand might lose the specificity. So rather than binding to this particular protein, it might go and bind to some other protein. And that is going to give rise to side effects. That's going to give rise to side effects that we don't want. That is why it is really essential for us to check this particular domain. And there is also another utility to this particular structured prediction. I'm going to discuss that later. Okay. Now, apart from this particular thing, there is also another server that could predict the secondary structure. See here. So this is another server. Now, what it does, this particular server was actually meant for Chow and Fastman secondary structure prediction server. Okay, they are using only Chow and Fastman algorithm to predict the secondary structure. But what if this prediction is not ultimate? How could I say that this prediction is the ultimate prediction? Because there are several other algorithms, and everyone is going to say that yes, that algorithm, their algorithm is better. So it is always better to compare few algorithms than predict the structure. So what this does is, they have three different algorithms. They have the Chow and Fastman, they have the Gore, and they have the neural networking to predict the particular structure of a particular protein. So similarly, what we have to do, we have to simply upload the protein in a flat format. Flat format means we can't use the FASTA format. Here we were using FASTA format, right? We're using the greater than sign and everything. But here we have to reject all of those. We only have to upload the protein structure in single letter amino acid code. Only the protein structure, nothing else. Okay. So after we upload it, then you have to simply select the which method do you want. I have selected all the three methods to simply check and cross check. Or you can use the sample input here. There's always a method that gives a sample input. Okay. And then simply click on start prediction, and they're going to predict the entire nature. So now, since I've used three different methods to predict the result, they are using three different methods to predict the result. So see, according to Chow and Fastman, it is predicting that this particular domain is alpha helix. So if I could compare here, see, this particular domain was assigned alpha helix, and this particular domain, starting from LLAY, this particular domain was assigned the beta sheet structure. So here too, we could find the same thing. Starting from this LLA, this particular domain is assigned the beta sheet structure. That was according to Chow and Fastman. But after, what about the Gore? The Gore is stating that this entire stretch is alpha helix, starting from here till here. The entire stretch is alpha helix. Whereas the neural networking is also predicting that the entire stretch is alpha helix. Now, combining these three methods, these three algorithms, they are going to give you the final algorithm, that this particular method or this particular region till this, the entire stretch is alpha helical region, and this particular domain is coiled, this particular domain. So you see, only if I'm using only the Chow and Fastman, it was stating that this particular domain has the tendency to form beta sheet, and as a result, this particular domain was assigned the beta sheet region because structurally that was the most favored. But according to Gore and neural networking method, we could see that this entire domain, they are also assigning this particular domain to random coil and all, but at the end we could find that this entire stretch is alpha helix. So this is obviously the much more better method. Why? Because this is comparing three different algorithms to give you the final result. Okay. And obviously it goes on like that. It simply tells you and gives you the idea that which part is alpha helix and all. It's the final domain. Okay. And it gives you the idea. So obviously what is better is obviously using this particular server actually gives you the more enlightened idea, because this actually uses a lot of different algorithms, and this is much better. Now obviously you might want to know that which is what method, why is what is giving. Now see, a basic principle of this Chow and Fastman is actually given here. The Gore method is also given here. I'm going to discuss the Gore and neural network, this method, how they predict and all in the later part. Okay. So this is how we could actually find and predict the secondary structure. Mind it, only the secondary structure of the particular protein, not the 3D structure. Okay, any question till this point?
No, sir, it's fine.
Okay, fantastic. Okay. Okay. So now we are diving into the 3D structure prediction. Okay. So what we have is first, now when we try to predict the 3D structure of the protein, there are 4 different methods. There are 4 different stages actually for predicting the 3D structure. The first stage is the backbone prediction. Now what is backbone? What is backbone of a protein?
It's the peptide bond that is formed.
Yes, the peptide bond connected with CONH bond. Okay,
Yeah.
removing the side chains, only the basic backbone is the backbone prediction. So that is the major thing. Why? Because that particular backbone is going to tell you whether the particular part is alpha helix or beta sheet or how the protein structure is going to look like. Now there are 3 different methods
Okay.
for backbone
I
prediction.
mean, so that is. So just one second. I'm sorry for the interruption.
Yeah, yes, yeah.
Whether it's alpha helix or beta sheet,
Yeah.
that is dependent on the backbone only. I mean, it's not dependent on the this thing side chains.
No, no, no. They are not dependent on side chain. Not generally dependent. Sometimes side chain also help you to predict. See, this is the general the backbone, the R1, R2. These are the side chains. Okay, this is the entire primary structure. So sometimes what happens is that this particular the side, this amide free amide bond and the carboxylic group, they actually start to form the hydrogen bonds. Okay, it starts to form hydrogen bond. Now, wherever there is I2, I plus 1 or I2, I plus 4 connections, then it usually forms the alpha helix structure. And when there is I2, I plus 5 connection, they form the beta sheet structure. The R groups or the side chain, they generally do not participate in the secondary structure formation. Okay, this secondary structure formation is purely a part of backbone. It's purely a part of backbone. The secondary is the side chains, they don't have a say over which structure is going to form and all. Okay, but stating that, obviously sometimes it is found that some of the amino acid or the side chains, they have a slight tendency to what should I say? They have a slight tendency to simply help the backbone to predict a particular structure. Okay, but that is very low in number. That is really low in number.
Yeah.
Okay. So there are 3 different methods for backbone: the ab initio method, the homology modeling, and the protein threading. Now, ab initio method means starting from the scratch. What is starting from the scratch? Starting from the scratch means they are not using any homologue of a particular molecule. But when I say a homologue, what is homology modeling? Now, see, as I've told you that there are various proteins whose structures are already solved. Right? Now, when a particular method or the structures are already solved in a particular system, then what happens is that they take the clue from there. So, for example, if I simply ask you today to draw a picture of a person, or draw a picture of a man or a woman, or draw a picture of a human, then you already know that how a human looks like. You know that they have 2 hands, 2 legs, so you are going to draw a picture of a human. But if someone doesn't have an idea that how a human looks like, then what? Then they have to solely base upon the genetic structure.
Ab initio method.
Yeah, that is the ab initio prediction. When you have no idea of how a protein or a part looks like, that is the ab initio prediction. So how does a homology modeling works? Now, this particular site, it harbors a lot of this particular PDB. This is a database where all the solved structure of a proteins are dumped or deposited. Okay, all the proteins whose structures are solved, they are deposited into this particular database. Okay, now as long you can see, there are 189,436 biological macromolecule structures that are deposited over here, and obviously maximum of them are proteins. Some of them are RNA structures and some of them are DNA structures. So obviously, when you dump all of the structures, for example, let me type a protein, suppose a VEGFA. Let me type in a VEGFA of a snake venom. Okay. Now, when I'm talking about the VEGF protein of snake venom, you can see that this is a particular crystal structure of the venom. This is a VEGF from the snake venom. Okay. Now, this particular structure is solved. How it is solved? It is solved by X-ray crystallographic method. Okay. X-ray diffraction method. The organism from which it has been divided is a Vipera aspis. This is the snake viper. Okay. And this is the protein. This is the VEGF protein, vascular endothelial growth factor. Okay. This is a toxin. Now, like this, several molecules or several protein structures are deposited over here. So when I am uploading a protein structure for 3D structuring, what they do is they take the help from here. I have uploaded a sequence. Obviously, for protein structuring, I have to upload the sequence. So whenever I upload a sequence, the inbuilt algorithm or inbuilt in the server, what they do is they come back to this RCC PDB website and they simply try to see how this particular, or if any protein stored in this particular database has sequence similarity to my queried protein. If they have any sequence similar to the queried protein. Okay. Now suppose suppose they have 3 different methods. Okay, there are 2 different structure prediction methods. Now, suppose they found out, yes, I have in this particular domain, I have a particular sequence. Suppose let me talk about this. Suppose I have this particular structure. Okay, this is MIAQLLAYY. Suppose this is the particular domain. So it is going to analyze all the protein structure to find out if any protein has similar kind of sequence there or not. And if they find that yes, there are a few proteins, suppose this VEGF protein, this has that particular same sequence, the same domain. So now it is going to take a look into this particular structure to see what structure have been assigned to that particular domain. What structure have been assigned to that particular domain. And then they assign the same structure over to this particular protein. Okay. Now, you might say that always it is not possible to find that. Yes, you are going to say, no, it's not always possible. So, for example, this VEGF protein, suppose this VEGF is also found in human, it's also found in plant and all. So, suppose the VEGF-A of human is not been structured, okay, but VEGF-A of the plant is structured. Now, the VEGF-A of plant and human is going to be different, but there are going to be certain similarity. There is going to be certain homology between them, right? So it's going to understand and find out the rate of homology, the percentage of homology, and it's going to, if there is same sequence, it's going to assign the same model to that. Okay, and that's how it predicts the entire secondary structure. Okay, so that is the homology modeling. Okay, and the next part is the protein threading. Now, protein threading is actually the best method for backbone prediction, but not every software does so. So there are few server, only one server that actually uses protein threading method. So what it does is in protein threading, what they do is they functionally cluster the entire thing. So I'm going to discuss protein threading later. Okay. But what it does is it actually clusters the entire protein. Suppose by protein structuring, I might find that this particular protein can be assigned 10 different structures or 20 different structures. So they form a cluster to find out which batch of structure have the minimum energy, right? Because the protein must have a minimum energy for protein structuring, right? I discussed in the last class, right? So when they have that minimum energy, when they find out that this particular protein or this batch of structures have lowest amount of energy, then they select only that cluster, and from that cluster they try to figure out the average structure. So that is the basic, what the protein threading is, and this is the best method for protein structuring. Okay. So after the backbone is predicted, what then they try to model the loops. Why? Because the loops are very difficult to model because they don't have a proper structure. So the loops are actually connected to one another. They are connected to different secondary structures so as that there is least amount of clashes. There is no clash among the different side chains and all, and so that the particular protein might have a proper folding. Okay. And after it is done, then they focus on the side chain packaging, because the R groups, as I've told you, that they have no, they do not participate in backbone structure prediction and all,
Second
but
one.
they have a very important role. What role? The role is that they can't simply point anywhere. They must be packed properly because the side chain volume is huge. There are few amino acids that have a huge side chains. So it is essential to see if the side chains are pointing outside or if the side chains are buried inside. Why? Because the ligand molecules, they are more prone to bind to the side chains. If we consider
Side
protein-protein
chains. Okay.
interactions, yes, if we consider protein-protein interaction or protein ligand interaction or suppose protein DNA interaction, then generally all these interactions are modulated by the side chains. The backbone have no tendency in or they have no part, they have very less part in binding to a ligand or to a protein or to a DNA structures. There the side chain plays the major role. So they have to be packed properly. And generally how does it happen? Generally the hydrophilic amino acids are projected outside. There are obviously few hydrophobic amino acids that are also projected outside. And that is what the side chain packaging means. That which amino acid is going to be projected outside, which residues are going to be buried inside. Okay, that's the side chain packaging. And it also ensures that there is no steric clashes between two side chains, because the side chains simply float around, right? So suppose the arginine and lysine, they also, suppose there is arginine and lysine that are just one after another. Now we know that arginine and lysine both are positively charged amino acid. So it might happen that since they are both positive charge, they might simply repel one another. So if they are not packed properly, they are going to repel one another and the structure is going to be destabilized. So it is very important to pack that particular side chain so there is no steric clashes, there is no electrostatic clashes as well. And finally, what you have the structure refinement part, where the entire protein structure is then refined, and energetically it is, what is refinement? Refinement means to see whether the hydrogens have been added or not. If there is no steric clashes, they are energetically, they are actually minimized so that you get a proper structure. Okay. So this is the ab initio modeling. That means that we are starting the entire thing from scratch. We have no idea. We don't want to, because sometimes you might find, suppose today we have discovered a protein or a bacteria from Mars. Now, obviously, if I bring a bacteria from Mars, it is not going to have any similarity with the amino acid present in India, in Earth, right? It's not going to have any similarity. So there we can't use homology modeling for structure prediction. So there we have to solely use the ab initio modeling. Similarly, suppose today I try to find out a particular organism. Which organism? Any organism. Suppose our organism found in the hydrothermal vent. Now, obviously, in hydrothermal vent, the temperature raises or it simply goes up till 10,000 degree. Okay. Now, obviously, in that particular domain, if some organism is alive, they also have the protein. But in our case, what happens if the temperature outside goes up, if our body temperature is not maintained at 37 degrees centigrade, then the enzymes or every protein, they are not prone to be properly folded. That is what happens. And what happens? They lose the function. And that is why what happens when we have a tremendous heat outside and we eat a lot of food, then the enzyme actually gets unfolded. And that's why we could see that in summertime, people are more prone to get stomach ailments. If they take a very oil rich food, it is really difficult for the digestive enzymes to digest. Why? Because the heat goes up and if the body fails to maintain the body temperature 37 degrees centigrade, then the protein gets unfolded and you get series of loose motions and all. But the same thing, when the particular animal is present in hydrothermal vent, then the protein is not unfolding. Why the protein is not unfolding? It is able to withstand a temperature of 100 degrees centigrade. It's able to withstand a temperature of 200 degrees centigrade. So how are those protein are able to withstand that temperature? Because they have some other method of folding, right? So this is why. So obviously, if I want to model that protein, I can't use the homology modeled protein, because generally all the proteins that are structured, that belongs to either to human or to plant or the virus or bacteria that thrives in 37 degrees centigrade. So in that particular aspect, we have to use the ab initio modeling. Okay, because in ab initio modeling, They simply use the amino acid chemical structure and physical structure for a proper structure prediction. Only the amino acid structure, nothing else. Okay. And in the previous class, we discussed the amino acid structures, right? Clear? Okay. So similarly, what they do is they
Yes.
simply use those amino acid structures to find out which amino acid has high propensity to form alpha helix, and they assign that role to that, and that's how they predict the structure. So now we are going to focus on to the okay. So this is one of the server that allows to you want to predict the 3D structure of a protein. Okay. So this is the Robeta, or this is from Baker's lab. Now obviously for first time use you have to register to this particular site. Okay, registration is really easy. You have to simply click on, you are going to find out a please register option. Just simply register, give your name, email ID and all. It's going to get registered. Okay. Now what is this? This particular protein structure method. Just give me a minute. Yes. Okay, so this particular server Roveta, this actually allows you to predict the 3D structure of any protein. You have to only input the sequence of that particular structure. But why it is very much used? Because this particular server is continuously evaluated by the Cameo. There is a board that continuously evaluates whether this particular server is able to maintain its quality. Every time a new algorithm is developed, every time some people finds out new, some researcher tries to, or finds out something new about the protein structure, some new information about protein structure, or the algorithm is refined, or the mathematical modeling is refined, they incorporate those into this particular server. As long as you can see that till August, the latest update was on August 2019, 2021, where they updated the browser. Okay, there's sometimes there are issues that crops up, and you could find out that recent bug fixes are continuously going on. Whenever there's a bug fix, they simply try to avert that and they focus on this particular thing. So what does it do? Actually an automated tool for protein structure prediction analysis. And for structure prediction, sequences submitted to the servers are actually passed into putative domains, and structural models are generated either by using the comparative modeling, that is homology modeling, or by using the ab initio modeling. But recently they have developed two other deep learning based methods, that is Rosetta Fold and TR Rosetta. Both are actually same. Rosetta Fold acts much better than TR Rosetta. Okay, I'm going to see and discuss. And after the structure prediction, if a confident match to this particular known structure is found, then they use the BLAST, SciBLAST, or 3D July. Okay, and then it is used for the template. If they find a particular method, then they use that as a 3D template, and they predict the structure. Obviously, if it is only for homology modeling. Okay, and if there is no match that is found, then the structure predictions are made by using the ab initio this Rosetta fragment insertion method. Okay, now for this particular method, the more important thing is that you can obviously use the experimental NMR constraint data that can also be submitted with a query sequence for this Rosetta NMR de novo structure determination. Okay, now there are also other methods, or what is that there are domain prediction method and molecular coordinates of models that spans the full length query and that are given as a result. And the server, as I've told you, they can use the NMR constraint to give you the proper structure and all. And since the multi-domain proteins are often difficult to crystallize and many are too large for NMR structure determination, this domain prediction using ROBETA, it can actually aid the structural genomics that are referred by expanding the pool of target from which the structures can be determined. Okay, and so that is why this particular method is really very good for structure prediction. So how to use it? So as you can see, there is two different methods. So first you have the structure prediction option over here. Click on it and then click on submit. Okay, so once you do it, the entire panel is going to open up. So the first and the most important that you have to do is the target name. So you can simply give any name, suppose a demo protein or something. And suppose now next what you have to do is you have to upload the protein sequence. Now suppose let me update this protein sequence as well. Now though the structure of this is already predicted, but still I'm using that as a model, and I am simply, let me edit the protein structure. So I'm simply doing this so as the protein structure changes, the entire sequence changes. Okay. So suppose this is complete a new protein that I now have. So you have to simply upload the protein sequence. Okay. And the major thing is that if you hover your pointer to this question mark, they are going to give you the idea that what each subset means. It's a protein sequence. What you have to do, you can upload your protein sequence in single letter amino acid code from 27 to 1000. But if you are using homology modeling, then obviously you can give protein sequence up to 2000. The amino acid sequence can be up to 2000. But for Rosetta Fold, it is only till 1400. Okay. So all this is done and all. Next is the, or you could also submit a particular molecule using the FASTA format. If you have a protein structure that is developed or sequenced in the FASTA format, you can upload it here. But obviously, I try to simply submit it over here. The next thing is that there are 4 different methods to predict. The first is the predict domain. Now, what the predict domain does? The predict domain, it is actually used to predict the protein domain, not the entire structure, but to predict the domain. Now there are various domains that are found in the protein. Can you name one?
Domain in the sense for my protein that I used in my PhD, it had 2 domains, one sensory that used to sense the light and one regulatory domain like the B7 domain which used to bind the DNA. Like this, are you like trying to say these are the different domains of my protein?
Yes, actually, very good. Generally, what a domain is any region of a protein that acts as a self-stabilizing molecule. Okay, even if you don't take the entire protein, if you take that particular domain can fold accordingly. Okay, and it folds independently from the rest. Yes, and it is a functional part, and it can function as well. Okay, that is a domain. So suppose if I want to, as you have told that you have a light sensory domain and all. Similarly, there are some proteins which have ATP binding domain. There are few proteins that have NADPH binding domain. For a zinc binding, if there are proteins that are metalloproteins, there are zinc binding domain. For hemoglobin, there is a iron binding domain, right? There are different domains. So if I want to find out the domains of my molecule, then I'm going to select the predict domain structure, because see, the ATP binding domain, whatever the protein it might be, the ATP has a proper structure, right? So obviously, whenever the ATP binds, that particular ATP binding domain of that particular protein is going to have similar structure, because the ligand has a proper structure, right? They have a single structure. So the ATP binding domain of all the protein, that particular protein might be of plant, it might be of a dog, it might be of human, it might be of a cat. That particular ATP binding domain is going to have a same structure throughout. That same thing is going for every other domain as well. But sometimes not every other domain, because for light sensing domain, as far as I know, I'm not expert in this, but there are 2 different methods. There is one domain which can predict only the black and white light. There's another domain that can actually assign colors to the entire structure, right? So for light sensing domain, there are different structures depending on how you see the light. There are a few animals which only visualize the light through as a black and white. So for them, they only have the black and white domain. So their domain is slightly different than that of human and all. Okay. And for the plant also, that is different because the plant are also able to photosynthesize using the tube light also, right? Light from the tube light. So their sensitizing domain is completely different. Okay. So if you want to predict the domain of any structure, then you have to click the predict domain structure, and that's all. Next is the AB. AB is the ab initio modeling. Okay. Ab initio modeling means, suppose, as I've told you, if I have a protein for which I already know that I have no homologs. This protein have no homolog whatsoever. So obviously, if they have no homolog, I'm going to avoid the homology modeling and I'm going to predict the structure as an ab initio modeling only when I'm sure that this particular protein has no homolog. Okay. And this is generally done for the molecules or the people who uses the high heat sensing proteins, or the proteins which are stabilized in the vacuum, and all the astrophysicists, they use this particular thing. For comparative modeling, as I've already discussed, this is the homology modeling. If you want to predict the structure by homology modeling, then obviously you're going to select this. Now generally the protein structures are solved by most of the protein structures solved by homology modeling. And if you might ask that which structure gives you the better idea, homology modeling or ab initio modeling, I'm obviously going to say homology modeling, because ab initio modeling, the algorithm, now in our body the protein folds in a ab initio modeling way. The protein folds in the ab initio modeling way. They don't use homology in our body. When the protein folds, it completely uses and only uses the amino acid sequence, amino acid structure, and when I say structure, both physical and chemical structure for protein folding. So ab initio method is the best method to predict the structure, but the algorithm or the mathematical modeling that we have for ab initio modeling is not as developed as the homology modeled one. So that is why for better structure prediction, it is obviously better to use the homology modeling. Though the body, in our body, we fold according to the ab initio method, but still we are unable to deduce the entire chemical functionality of the amino acid. We know how amino acid looks like, what is the chemical structure, and it is a small molecule. But when that particular amino acid is in conjuncture with all the other amino acid, then how it is behaving, then how the mathematics is going to be written down, because all the algorithms have to be written down mathematically, right? So that particular part is not yet that much developed. So once we, in the near future, if we find out a particular phase that where we know that we have found out everything, the entire protein structure is unknown to us, then obviously the protein is going to be found, or it is going to be folded ab initio method. Okay. So that is the comparative model. And next we have the TR method and the Rosetta fold. Now both of them are same. They both uses the deep learning method. Now what is that? Now each day, every day, thousands of people submit their sequence into this particular site, right? They submit their protein over here to try to analyze or find out the 3D structure. Now when they are doing this continuously, then, and obviously it is going to give you a model. The output of this entire structure, they gives you 4 models, and you have to choose according from the 4 models, you have to choose your best structure. So every day thousands of people, researchers submit the sequence, they get the structure and they choose the structure. So this particular thing is remembered by this server. The server or this algorithm, they remember which scientist or which researchers are submitting which sequence, and if that is the sequence probability, then which models are being selected by them. Okay, so they remember it and they use all those knowledge for protein structuring. Suppose today I've modeled this particular protein and they give me 4 models. So in one model I have this particular domain as alpha helix, suppose. In the other 3 models I don't have this particular domain as alpha helix. Okay, so after analysis, suppose I find out that this is the best method or this is the best structure, I select this structure. So it is going to remember that this particular sequence was assigned alpha helix structure in one model, and the rest of the 3 model, this was not assigned the alpha helix structure, but a scientist choose this model. So that wise they remember that yes, this particular model should have an alpha helix domain. That is what people are selecting. Now, this is not a single, I'm giving a single example, but every day thousands of people are submitting it. So they take all their cumulative information, it analyzes all those information, and then it uses those information to predict and fold your protein. So that is the Rosetta fold, and this is perhaps the best method currently for protein folding. But still, there are few people who are skeptical about it, and they prefer to use the homology modeling, where they simply go to the RCSB PDB website, they simply use the cluster W format to align all the proteins. Suppose there are 1,000 proteins, they align the 1,000 proteins to your structure, try to find out the homology, and once the homology has been found out, they try to assign that particular domain to that particular protein, and that's how they sequence the, or they give you the structure. Okay. Now,
So
yes.
yeah, can we like do it? You told us we can align, for example, 1,000 proteins of which is like similar to my protein of interest using the cluster W pattern. So can we do it like directly from PDB, or we have to download individually all the sequences and only then we can?
Yes, you have to download the individual sequences and then you have to do it manually.
Okay.
But you can do it by writing a Python code, but not everyone knows that particular how to write the code and all. You can do it by writing a Python code, and it requires it's a very long procedure, but it's generally advised to, for generally the biologists, what they do is they manually download the sequence and they have to manually align it, and then they have to feed it over it. And there is also option for that. If you do that, then you already know which is your homology, right? Then you can simply enter. For competitive modeling, you can upload the PDB template. Suppose you know that this particular model, this particular molecule has high homology to this molecule over there. Okay, so this is 1WQ8. Okay. So what you can do is you are going to write down 1WQ8. So it is going to use only this PDB ID as your template. Okay, it is going to use only this PDB ID for your template.
This or what template, you can do, or
yes, as your template for homology modeling. Or what you can do, if you want to add multiple proteins, then you have to write down or save all the structures in a particular format, and then you have to upload that over here, choose file. Okay.
Okay, and will you teach that the coding part in this course or no?
No.
Okay.
So after it is done, because now generally it is also not required. Because this the servers, what they do is they are pretty efficient in aligning your queried molecule to the PDB website. Okay, they are pretty efficient in doing that, and they give you pretty good job. But obviously, if you know that, suppose I'm using a VGF protein, so I know that VGF protein has many other homologs, right? So at that particular time, I can simply use to give you the PDB ID and all. But if it is not known, then obviously advised to simply not to give anything over here, and they can automatically search the entire domain, because there are more than 10,000 or 1 lakh protein molecules that are stored over here, more than 1 lakh. So it is really difficult to analyze all those manually, right? So it is better to use that method. So yes, once you upload it, they are going to give you the idea. See, I have written down the PDB code, so they have taken that particular value and they are showing you which structure that you want. So that their algorithm is pretty much well defined. The algorithm used in this particular server is pretty much well defined. Okay. Okay. So that is, once that is done. Okay. So now the next thing that you again have is this open fragment panel, or you have the constant panel. So what are this constant panel and all? In this particular panel, suppose I have already performed an NMR spectroscopy and I know which particular domain is alpha helix, or this particular domain has a beta sheet region and all. I have performed the NMR of small domain. I know the idea. I have the basic idea. Then what can I do? I can simply assign that particular NMR constraint format over here. Okay, I can assign the NMR constraint format over here, so that, because obviously if I found out that through NMR, suppose through NMR I find out that this M is connected to this W by hydrogen bond. Okay, so I'm going to obviously let them know that this particular domain, this particular amino acid, or the alpha helix of this amino acid is connected to this amino acid via hydrogen bond. So make them connected. So if I give those constraints, it is going to apply those constraints for the protein structuring. So it is much more better structure that I get. Okay.
Okay.
And there is also a secondary structure upload fragment, where if you have an idea about protein secondary structuring, that you can obviously give over here. For example, suppose I have already found out that this particular domain has a alpha helix domain. Okay, suppose this particular molecule has alpha helix domain. So what I can do is I can write down over here. I can easily write down over here that this particular domain is assigned the alpha helix structure. So please use that particular domain over here. Give alpha helix nature to this particular domain, beta sheet to domain, or random coil to this particular amino acid. And if I upload the fragment secondary structure, then they are going to use this particular fragment. They are going to keep those fragment as it is, and then they are going to fold the rest of the protein with respect to that particular domain. Clear? So this is done when
No, sir. How do we write here? That is not clear.
yeah, I'm
How do
showing you
we
that.
write here?
Yes, I'm showing you that. Have you opened up this particular fragment? Yes, see, here you could see that for first what you have to do, first you have to write down the name of the sequence. Okay. Now remember, there are 3 coordinates, x, y, z. For coils, the x is 1, y, z is 0. For helix, x is 0, y is 1, z is 0. For alpha, for beta sheet, x, y, 0, z is 1. So suppose my first molecule over here is, suppose methionine. Suppose. So I'm going to write 1 methionine. It has been assigned a random coiled structure. So now I'm going to give. So this is the first. Suppose I want to assign second. Suppose I had cysteine. Suppose cysteine is alpha helix. So I'm going to write helix. Then helix, for helix, the y coordinate is 1. X and Z is 0. Suppose the third one was alpha helix, suppose beta sheet, suppose that was suppose tryptophan. So I'm going to write down over here E, beta sheet, 0, 0, 1. So like this you have to write down for the entire, all the molecules or all the amino acids that you have. Okay. You have to go on writing it down like that. And so while you write it down, they are going to make sure that this particular domain or this amino acid is assigned alpha helix structure, or this is assigned the random coil structure, this is assigned beta sheet structure, like that. Okay. And once it is done, simply write down what is 3 plus 2, 3 plus 2 equal to 5, and then simply click on submit. And it's going to submit. And now I have nothing over here, so that is why it's not showing. And since you register, and once you register, they are going to use, they are going to send you the mail. Sometime sometimes it takes time. Sometimes they are going to give you the structure in 6 hours. Sometimes they're going to take 2 to 3 days. It all depends upon the length of the protein, the load on the server, all these things. Okay. And after that, after they do it, they're going to send you a mail, and simply click on the mail. They're going to give you a link to download the structures. Okay.
Okay.
And then you have to simply select that particular structure and over here. So do one thing, simply upload a particular protein for your structuring. And the next class, what we're going to do is we're going to analyze that particular molecule. Okay, we're going to analyze the structure, because there are going to be multiple structures. So you have to select one structure. Generally, what they do is they give 4 structures. They give 4 structures and they're ranked accordingly. The best, first is the best one, second is the second best, third is the third best, fourth is the fourth best. But sometimes that is not the rule of thumb. Sometimes the fourth best structure seems to be the best structure. Okay. So that depends upon the researcher to analyze the structure and all. So in the next class, do one thing. we're going to discuss how to analyze the structure. Okay. So it will be better if you have a structure. Simply upload a molecule over there, and you're going to get the protein structure in 2 to 3 days, and then we can analyze that particular structure in the next class.
Okay, sir.
Okay. Any more questions?
No, nothing.
Okay. So simply submit, take your protein, demo protein, and simply upload a particular molecule, and you're going to get the job done. Okay. And you can use any of the model. You can use a competitive modeling or Rosetta Fold, any of that you want. But TR and Rosetta Fold both are same. but Rosetta Fold is much better than TR. So don't use TR. Okay. If you are going to use TR and Rosetta Fold, always select the Rosetta Fold.
Or I can select this ab initio also.
Yes, or you can select ab initio if you want. You can select ab initio. In fact, you can do is you can upload the same protein and you can select homology modeling and ab initio modeling to check how they are different. You can also do that.
Yes.
So I'm going to get an idea that how both structurings are done and all, how they are different. Okay.
Okay.
Yeah, sure. Okay. So thank you for this class. Yeah, sure.